everyone, welcome back to the 14th video of the 100-day biopharmaceutics challenge, whereby we are going to cover the determination and factors affecting protein drug binding. So let's see what exactly are these factors, but before that I would like to mention how this conversion is going to happen. Let's suppose we have talked about a drug which is going to enter into this systemic circulation. This is the blood fraction, and then it is going to be distributed into the multiple organs. This is how we have categorized the drug distribution. Now, what are going to be the major steps? These steps will actually be the part of the factors that are going to affect. Number one is going to be the permeation of free drug through the blood, right? Which we know that it is going to enter the blood component. This is going to be the interstitium part. This is going to be the interstitium part. Then the drug is going to cross these membranes through the capillary wall and enter the ECF, extracellular fluid, right? After it enters the extracellular fluid, or we can say it enters the interstitium point, now it is going to enter into the different organs. So that is going to be affecting the second step, which is going to be the permeation of drug through the different organs. So while you are talking about the permeation of drug through the different organ, it has to cross their membrane also. That is going to then enter into the particular organ. So in this second step, the drug is going to enter the intracellular part, right? ECF, it is trying to enter the ECF. Now this ECF, this crossing of the membrane will depend on two things. It will depend on the perfusion rate, that is the rate of, we can say, blood flow to that particular organ. Secondly, it is going to be affected by the membrane permeability, right? The membrane with the concept of like dissolve like will be happily letting the lipophilic molecules to enter, but if they have a larger sizes, then the entry is going to be a challenge. This permeability here, we can call it as the tissue permeability. That tissue permeability means it is going to be dependent on the type and nature and the properties of drug, including their molecular sizes. You already know that a size range more than 400 Dalton will be very difficult for the drug molecule to be able to cross the membrane, which we have seen in the drug absorption part also. pKa, based on the drug nature itself, whether it's ionized or unionized fraction, it is going to be letting it, only then it will be able to pass across the membrane. Then we have is the partition coefficient, which is a very good indication of the permeability. If the molecule is lipophilic with the concept of like dissolve like and in particular size range, then only it will allow it to pass across that. Then we have physiological barriers to the drug distribution. You all know in the case of barriers, we have blood-brain barrier, blood ECF barrier, right? Blood placental barrier. So blood testis barrier, these all barriers are going to restrict the entry of the drug in a particular organ. But all these factors can be actually conceptualized using the major thing, that is going to be a perfusion rate. To what organ how much blood supply is going to actually happen? We have highly perfused organs, moderately perfused organs, poorly perfused organs as well. In the highly perfused organs, you all have studied about the kidneys, lungs, heart. In the moderately perfused organs, you have covered about the skin and the muscle, and poorly we have fat and the bone, right? Then comes your binding of drug to the tissue compartment. We have already seen the drug either binds to the protein or either binds to the tissue compartment. That binding will be affected by its, that binding will alter its pharmacokinetics, its size will be altered, so as its property is going to be altered, as well as the binding of drug to the extravascular tissue proteins. Then we have miscellaneous factors. We have age, pregnancy, obesity, any disease condition, dietary state, any drug interactions that are going to follow. So majorly the whole point is, if the these factors are affecting the drug molecule, number one thing has to be a particular nature, size, range, so that it is actually going to enter the other organ. That means the tissue permeability and how much, to what extent it binds to the protein is very important. Only then it will be able to cross the barriers. For example, in this case you can see a drug molecule which is unionized this drug molecule will be actually able to cross this capillary wall and enter the extracellular fluid, or we call it as interstitium. After then it is going to cross the cell membrane. Because of the like dissolve like concept, it will have no issue in crossing the membrane and enters the intracellular fluid. Now after this let's take an example of another ionized molecule. This ionized molecule, if it is smaller in size, it will be able to cross the capillary wall, but it will again be stuck. It will not cross the cell membrane because of the nature as well as other limitations to it. So based on their ionization, based on their size and nature, their entry is very much going to hamper based. That is why we say partition coefficient, tissue permeability, we say log P values, ionization, all these are going to affect the drug protein binding. So what are the methods by which we can determine the drug protein binding? So it largely depends on the, are you going to separate the free molecule and the drug bound fraction, or you are not going to separate the free molecule and the drug bound fraction. So just see, we have two methods. We have indirect techniques and in the direct techniques. In the indirect techniques, the bound form will be separated from the free fraction. Let's take an example of the dialysis here. You all have seen that we actually take a dialysis membrane. In this dialysis membrane, we make a dialysis bag out of it, put our formulation into it. So, or we have, suppose, this dialysis bag right here. Okay, let's draw it here. So let's see. So in this case, the red molecules that I am drawing, this is a bound fraction. Let's see. This is a bound fraction. And the purple molecules that I have drawn will be your drug, free drug moieties, right, inside the dialysis bag. So what you are going to do, you want to purify it, you want to keep it just with the drug protein bound fraction, and you want to eliminate the free fraction. So this purification, you are going to just disperse this into a particular media based on the size of the membrane and the molecule. The free molecules are going to come out of this, and the drug fraction, which is just the protein bound fraction, will be remaining inside. So that can be quantified using the analytical techniques as well. So in this case, generally it is applied in the biological sample, serum, plasma, right, blood. So for the determination of percentage of binding, we have techniques, we call it as ultrafiltration, ultracentrifugation, dialysis, dynamic dialysis, equilibrium dialysis we have. In the direct techniques, these are do not require the separation of bound form of the drug. These are directly estimated using the fluorimetry, ion selective electrodes and UV spectroscopy, right? You can spot the differences between, from your exam point of view, the techniques, name of the techniques, and what is the basis. Are they going to eliminate the bound, or the they are going to differentiate between the bound and your free fraction or not? That is very important. Okay, I hope it is clear that how the factors are going to actually affect your protein binding, as well as what is the main determination techniques. Thank you so much.